So let's, this is final for this review and phases of response to an initial infection. So let's focus on the timeline, which is important for us to know. So when there's an infection happens, we need an immediate first line of defense as an innate immunity, that is a recognition by preformed, non-specific, and broadly specific effectors, such as we see LPS, then we recognize it and kill this, remove this infectious agents. So an early induced any innate response involves, let's say, from 4 hours to several days. Then now it's recognized recognition of this microbial associated molecular parent, then it causes inflammation. You remember that you've got, let's say, some hit or a small cut, and you have inflammation. So, and it lasts for several days. So this inflammation recruitment and activation of effector cells that's coming in, and then removal of infectious agents. And now after several days, let's say more than 96 hours, let's say about a week or after, now adaptive immune response will take part. So the infection and that transport of this antigen to go to the lymphoid organs such as lymph node, and then there's a lot of naive B cell and T cell. Some of them will recognize this specific antigen. So recognition and those specific B cell and T cell will have proliferate. We call it clonal expansion because they will generate exactly the same antigen, the same type of T cell receptors, and they will differentiate into an effector activated cells and B cell and T cell, and they will remove infectious agents like cleanly over time. So as a summary, so this engineering immunity section, we learned about the role of vaccines in the prevention of disease. And especially we study about the antibodies in the immune system and ways how to use antibody to prevent diseases and even treatment of disease. So that we learned the engineering of antibodies, like bare human ones into animal grown ones and humanized antibodies, etc. And so that we understand the antibody structure and different kinds of antibody classes. And we talk about the differences between monoclonal antibodies, which could be used for therapeutic purpose and research purpose, and also polyclonal antibodies, and how to manufacture, create monoclonal antibody, which led to Nobel Prize, and requirement for effective vaccine development. And finally, we have a immunology 101, or the basics of immunology, such as innate and adaptive immunity. Thank you for your attention.